So, good evening, doctors. I'm Shumon on behalf of Shield Healthcare, welcoming you all in today's session on Microbiome and ART, and our guest speaker is Dr. Aamani, madam. Before moving into the session, let's have a look on this Shield Connect platform. It's basically a knowledge sharing platform of Shield Healthcare. Here in the blog section, we will find different blogs written by many eminent doctors across India in various topics as well. And these are the awareness section in each and every awareness section. It consists of all the related informations like PCOS, menopause, GDM, and the other disease also. And these are the past webinar section. We have conducted the webinars in our platform till date. And the most important thing is our key opinion leaders. So more than 500 doctors are currently supporting us across India for an understanding initiative of Shield Healthcare. So I would like to request all the participants, please have a look on this website, as it's very important in terms of our subject area. Now it's time to start the session with Madam, and it's my honor to introduce Madam, Madam Rita MD, Gynec, and fellow in infertility. Madam is a senior consultant, Raja Hospital, Yusufgura, Hyderabad. So with this short introduction, I request Madam to take over the session, and meanwhile I request all the participants for your active participation, and please post your queries on the QA dashboard of the Shield Connect. So once the talk will be over, we shall have a short discussion on this topic with Madam. So thank you very much, Madam, for your valuable time once again in our platform Shield Connect, and over to you for further proceedings. Thank you very much.
Good evening to all. Hello. Hello.
Yes, Madam.
Yeah,
you are audible.
can I start my session?
Yeah, sure, ma'am.
Yeah, okay. So today's topic is, good evening, everybody. Today's topic is vaginal microbiome and ART. Very interesting topic. Next slide, please. The vaginal microbiome plays an important role in maintaining the woman's overall health. The vaginal microbiome community plays an important role in vaginal immunity, innate immunity, and the inhibition of all the three, bacterial, viral, and also the yeast infections. If you see in the humans, in the intestines or the GIT, the microbiome composes of 29% of the total. And in the urogenital system, it contributes 9%. Then vaginal microbiome is dynamic and changes throughout the woman's life and is commonly dominated by the Lactobacillus species, which we know is found in the curd. Next slide, please. Yeah. So what is this VMB or vaginal microbiome? Vaginal health is influenced significantly by this microbiome. And to human reproduction and public health, it has attracted less attention, basically. The vaginal microbiome is an intricate and dynamic microsystem that constantly undergoes fluctuations during the female, as a child, during the menstrual cycle, reproductive stage of the woman, and the menopause. The vagina harbors many microorganisms, which is called the microsystem. It contains many billions of friendly microbes. Data from, so one of the study done by Chen et al. In 2017 it's a new invention. 2017 was the one study done by Chen et al. He studied on 110 persons of reproductive age, and that revealed that vagina contains 10 into the power of 10 or 10 into the power of 11 bacteria. That is friendly bacteria, which we are talking about. The indigenous environment, microorganisms, and their genomes jointly compose the entire habitat, also known as the VMB or the vaginal microbiome. So let's go back to this sentence again. Vaginal microbiome contains what? The indigenous environment, the microorganisms, and the genomes jointly. The environment, the microorganisms, and the genomes. These combinedly form the VMB, what we are discussing today. And VMB plays a major role in ART. Next slide, please. Next slide, please. Yeah. So there is a very, no, please. Go back to the back slide. Yeah, thank you. There is a very gentle balance, which we are going to know right now. homeostasis of vaginal microbiome. So in this ecosystem, the homeostatic and the mutualistic relationship exists between these bacteria and the host, human host. The host provides a good environment which contains nutritious, humid, warm environment for the microbes to thrive on. And in turn, what does the microbes do? They produce antimicrobial and anti-inflammatory factors. Like it doesn't allow anybody else to come into that area. Thus, the first line of defense against non-indigenous microorganisms is established. So this is the natural immunity of the body to fight against the viruses, bacteria, and the yeast infections. So when we see there's a very delicate balance between the host and the microbiome. So what does the host provide? Host provide a nutritious, humid, and a warm environment for the microbes to grow. And what does the microbes do? They produce antibacterial, that is antimicrobial, anti-inflammatory factors. Very interesting, right? So next, homeostasis of vaginal communities. So that is a delicate balance or dynamic equilibrium between the host and the microorganisms. Maybe China and Pakistan. So yeah. So coming to this, homeostasis. Vaginal microbiota plays a pivotal role in preventing colonization by microorganisms responsible for genetic infections. So this is the first barrier or fur protection barrier which is formed, protecting the human body from the invasion of the microorganisms. So we can go for bacteria, virus, or yeast. The composition of the vaginal ecosystem changes in response to the exogenous and endogenous influences. We'll come to that. See, the composition, what we said, the humus-friendly environment created by the host is influenced by the exogenous and also the endogenous influences. That is from the hormones from the body, the metabolic state of the body, and also the environment. We'll come to that in the next slide. Next slide, please. Yeah. So this is a beautiful slide showing the balance between the harmful bacteria and the good bacteria. Vaginal homeostasis. The vaginal homeostasis, that is the good balance between the good bacteria and the host, will keep the harmful bacteria away. If the good bacteria concentration or the 10 to the power of 11 concentration is decreased, what will happen to the harmful bacteria? They'll have more power, or we can say they'll have the chance to feed on the nutritious fluid or nutritious thing, and the harmful bacteria will increase, which will cause BV, that is bacteria vaginosis. Next slide, please. So let us see what are these good bacteria that are good for our body. Okay, they have divided it into 5 groups basically, depending on the concentrations and the frequently found bacteria. All these bacteria are a group of Lactobacillus. Lactobacillus is nothing but found in the curd. Okay, so let's go into the group 1. This is maximally dominated, easily tripled. And the group 1 is Lactobacillus crispatus. 26.2% of the population is seen. That means they took the vaginal sample of around 1,000 women and they have isolated the bacteria which predominates and which doesn't. And they have also studied like how they reproduce and what is the time of replication of these bacteria. So depending on that, they have made into 5 groups. So the first group is Lactobacillus crispatus, and it goes up to 26.2% of the population. Group 2 is 6.3%, and it is Lactobacillus gasseri. It is a dominated one. Group 3 is the 34.1. This is Lactobacillus iners, And group 5 is 5.3% is Lactobacillus genesi dominated. And group 5 is classified as non-lactobacillus dominated and included. These include Gardnerina, Prevectera, Cornybacterium, and Aptobium. Okay. So next slide, please. These are just the names, like how we must be remembering our botany class when we were doing our BIPC, naming the plants. So we are naming the organisms, like Lactobacillus species. Next slide, please. Yeah. So we have 3 phases in our life. That is, a woman has 3 phases in her life. That is the premenstrual period, menstrual period, and a menopause period. So the hormones fluctuate in these 3 periods of time. In childhood, there is no estrogen, there is no progesterone in a child. In the reproductive age, the estrogen, progesterone, they play role for the regular menstrual cycle. And during the menopausal phase, again, there is loss of estrogen and progesterone. So how does estrogen and progesterone play a role for our microbiome? Let's go for this. So basically, the vaginal pH has to be maintained at 4.7, 4 to 4.7. So it is maintained through this lactobacilli. How does this happen? Let's go through it. So let's read the slides now. High circulating levels of estrogen and progesterone. Lactobacilli dominance in vagina. Estrogen, what does it cause? It helps in the deposition of glycogen in the vaginal epithelium. Got it? This is a very important point. So then the estrogen, when the estrogen hormone increases in the body, it causes deposition of the glycogen. We all know what glycogen is. It's a sugar, right? So glycogen in the vaginal epithelium. So there it goes, right? Unless we have Swiggy, the food won't come home. Similarly, now lactobacillus, what it does is it breaks down the glycogen into lactic acid with the presence of progesterone. Okay, so when the progesterone is there, the lactobacilli or microorganisms, which we are reading about right now, will break this glycogen and release lactic acid, which will cause the acidic pH in the vagina, that is 4. And as we know very clearly, the bacteria, the virus, fungi cannot live in an acidic atmosphere, right? So this is the beauty of nature, right? Now it's very clear. So we have estrogen and progesterone in our body. Estrogen deposits the glycogen in the vaginal epithelium. The progesterone is released and our Swiggy, that is lactobacilli, will come down and break the glycogen into lactic acid and will maintain the pH at 4. Good. Interesting, right? Next slide, please. Okay. So now I was telling you about the pHs. Let's see how our body maintains the pH in different organs, like our reproductive organs. So microorganisms are present not only in the vagina, but also in the upper reproductive system, including ovaries, fallopian tubes, and the uterus. So this is a very beautiful slide here. So now vagina, it is 4.4 to 4.7. See, when we come to cervix, it's more acidic, 7. The pH is maintained at 7 there. And when you move up, uterus, it is 7. Fallopian tube, it gets slightly more, 7.94 or bargain 8, right? So the lactobacillus, firmicutes, bacteroidetes, proteobacteria, acetinobacteria, these are friendly bacteria. So E. coli and anaerobes predominate in childhood. Lactobacillus colonization begins in puberty and decreases after menopause. As I said, right, we need lactobacillus feeds on what? Glycogen. Glycogen is formed only when there is estrogen. Enteric and anaerobic bacteria are co-dominant in postmenopause women. So the postmenopause and the childhood, 55% to 75%, the organisms have a good, humus for growth. When it comes to the reproductive period, menstruation starts, puberty starts, the pH drops to 4.7, and many bacteria are controlled by this mechanism. Next slide, see. Next slide, please. Yeah. So MVB and ART. MVB is vaginal microbiome and ART. That's our topic today. So let's come to it. Now we know what microbiomes are. Now we know what microbiome is doing in the vagina. And we have come to its effects on ART. First slide. Then the female reproductive tract has an active microbiome, and it is suggested that these microbiomes could influence the outcome of ART. They definitely do influence the outcome of ART. Many studies found an abnormal vaginal microbiome had a negative effect on ART. An abnormal vaginal microbiota has been associated with a poor reproductive outcome in patients undergoing IVF, IUI, natural treatment, as well as early abortions, preterm labor, and in IVF treatments. Studies confirm that a vaginal microbiota rich in Lactobacillus species without bacterial vaginosis, either clinical or subclinical, leads to more positive outcomes with ART. This is a proven study. Many studies are there on this topic. Babu et al. 2017, he studied around 1,000 members. Eckert et al. 2003, Mangot Bertland et al. 2013, Moore et al. 2000, Haar et al. 2016. This is a person who performed a prospective study on reproductive age of women specifically undergoing IVF. Kujral et al. 2019. It's a recent study. He studied around 850 patients who were undergoing IVF. What he did was he took a vaginal swab and a pap smear for all the patients who were undergoing the IVF treatment, and found that embryo implantation was less successful in women with reduced Lactobacillus species in the vaginal microbiota. Very interesting study. Next slide, please. So VMB and pregnancy. The microbiota in pregnancy vagina is less rich and less diverse as compared to non-pregnant vagina, with a predominance of Lactobacillus species. The significant higher stability of the VMB in early stage of pregnancy and the same increase with gestational age. We know, right, in the initial stages, the progesterone is high. As the pregnancy goes on, the progesterone levels decrease and the estrogen levels increase. So that is what he says, early stage of pregnancy and the gestational age. Etiology, including estrogen increasing the thickness of the vaginal mucosa, thereby increasing the glycogen deposition. The glycogen deposition acts as a chemotactic agent by microbes, as this is a major substrate used by these microbes, which is broken to glucose and then fermented to lactic acid, thereby playing a significant role in lowering the vaginal pH. We have gone through this point in the previous slide. Next slide, please. Yes. So the main function of the vaginal microbiota, that is our friendly microbiomes. What do they do? Infections. We have seen right now, Lactobacilli microflora disturbed. When this is disturbed, that is Lactobacillus are disturbed. The pH is greater than 4.5. The organisms, they invade, the yellow, white, grayish discharge, inflammatory changes, Gardnerella or mobilinus. Next, aerobic. Lactobacilli are disturbed. The pH is 6 to 8. Yellowish discharge, Staphylococci, E. coli. So next, coming to the other groups, the ethnic group, that's changes in pH. Lactobacillus colonization depends on the ethnic group also. This depends on the genes also, right? Ethnic group. Others, smoking. Smoking, when smoking is there is decrease in the estrogen levels. So presence of BPDA in the vaginal secretions, decrementation of Lactobacillus species, the pH is altered. Cancer, depletion of Lactobacillus species, high diversity of the microorganisms. So with age, As we studied this, depending on the hormones, that is in childhood, there is no estrogen, there is no glycogen deposition in the vaginal epithelium, very low lactobacillus species, high pH, prone for infection. Adult women. So this is a reproductive age where there is good estrogen, good glycogen levels, thick vaginal epithelium, good species, good number of lactobacillus species, low pH, secure. Post-menopausal. Again, the estrogen levels fall, glycogen level fall, thin and disrupted vaginal epithelium, low lactobacillus species, high pH, prone for infection. And coming to the pregnancy, lower concentration of the mycoplasma and uroplasma, decrease in pH, increment in vaginal secretions. So in the pregnancy, only difference is the secretions increase. Next slide, please. Which does not have any effect on the VMB though. Okay. Altered vaginal microbiota. What does it cause? Same, BV, bacteria vaginosis, vulvovaginal candidiosis, UTI, PID. So we need our friendly bacteria. Swiggy. Next slide, please. Coming to the lactobacilli. Lactobacilli provides a defense against the pathogenic colonization, such as all the organisms, that is trichomoniasis, candida, gardnerella, and by producing lactic acid and oxidation radicals, bacteriocins, and modulating the non-specific host immune response. They survive under low gastric pH, bile salt and passage to the intestine, and then ascended through functional intervention into the vagina. Same principle is applied even in the GIT. Next slide, please. In our intestines. So coming to our topic, IVF outcome. So lactobacillus dominant microbiota in the vagina, there is increase in the IVF outcome. Lower implantation when there is less, this is the cutoff point of the lactobacillus species, that is 10 to the power of 11, 10 to the power of 12. If the vaginal microbiota are less than 10 to the power of 10 approximately, then the IVF outcome is lower implantation rate, increased number of miscarriages, decreased number of term pregnancies, reduction of pregnancy rates. What we notice is higher concentration of Gardnerella vaginalis, Atropodium vagina, and low abundance of lactobacilli, high abundance of E. cocci, Enterobacteriaceae, Streptococci, Staphylococci, gram-negative bacteria, and low abundance of lactobacilli. The patients with lactobacillus dominant microbiota, that is 80 to 84 level of concentration, the success rate has increased. While lactobacillus species have been reported to exert beneficial effects, the specific endometrial or vaginal dysbiosis, that is less good bacteria, have been related to worse success rate, and references are reported to be failure, IVF failure. Next slide, please. Mechanism of urogenital colonization. Hematogenous spread of bacteria. So uterine microbiome, fallopian tubes, ovary, pouch of ductless, vagina, peritoneal cavity, endometrium. From anywhere, even from the blood system, the bacteria can attack the reproductive system. Next slide, please. Okay, this is very good. See, absence of vaginal lactobacillus. So the Nugent score, that is good bacteria, is 0 here, whereas the good bacteria is 10 here. See, in the second slide, it is 10. The first slide, it is 0. In the first slide, we seeing rod-shaped bacteria, gram-positive and rod-shaped bacteria is dominant here. That is bacteria vaginosis. And when we see here, Nugent score is 10, where we have good bacteria with us, Lactobacillus, and the rod-shaped bacteria is almost nil here. So overgrowth, often asymptomatic overgrowth, gram-negative or variable anaerobic bacteria, Gardnerella, Atropidium, Pervidia, Cnidia, Megaspira, symptoms. Odor, thick vaginal secretions, and clue cells are seen, and the pH is greater than 4.4. Next slide, please. Vaginal pH is a very simple test which will tell us the vagina is healthy, has good bacteria, or is dominated by the bad bacteria. So here in this slide, so the VMB composition, we have major ethnic groups in Amsterdam and Dutch background. They have, studied different people from different background. That is the total population: Dutch, South Asian, African, Ghanaian, Turkish, and Moroccan. So these are just different species of the Lactobacillus, and they have basically studied the incidence and different ethnic background or the genetic role in VMBs. Next slide, please. So pro and anti-inflammatory effects of vaginal bacteria. So this, what they did is they have inoculated the good bacteria or the VMBs in the vagina, and they have studied the CD4 T cell counts, the lactic acid levels, and the pH levels to see how even they're acting on the bacteria. On what they have found is CD4 T levels are elevated in women with high risk bacteria, and lactic acid at low prevalent production of inflammatory mediators. So the mediators are decreased when you infiltrate the VMBs. Next slide, please. Lactobacillus dominance and low pH. These are the studies just to, they established or they proved that when the bacteria Lactobacillus is there, the pH is less. So the predominance of Lactobacilli, 4, 5, and the amount of pH. See the 4 pH. Humans. Human pH 3.5. Apes and monkeys, and these are just studies what they conducted. Next slide, please. Glycogen levels in the vaginal lumen are variable. Postmenopausal, the glycogen is less. Prepubertal, glycogen is less. And macacus, that is low, and we have seen that low Lactobacillus is there when the pH is low. Next slide, please. Now, coming to this is good. A meta-analysis of bacterial vaginosis and infertile population. This is right, what they took. The patients would not have kids. They took them around 1,000 to 2,000, 5,000, I think so. And what they noticed is a meta-analysis of bacterial vaginosis. Prevalence of bacterial vaginosis to be 19%. Asymptomatic is up to 50%. Notice that the incidence of BV asymptomatic is 50%. And it is linked to preterm birth, infections, abortions, surgical abortions, negative impact on female fertility, infertility issues, tubal factors, etc. Next slide, please. So the incidence of bacterial vaginosis in population is about 50%. That is quite high. Effect of ART on vaginal microbiota. Right, VMB and ART. That's our topic, right? Circulating hormones impact greatly on the VMB, that we have noticed. Estrogen and progesterone. And indirectly susceptibility of the woman to infection. Exogenous hormones have an important impact on the vaginal environment and the composition of the VMB. Next slide, please. 4 months of GnRH treatment. This is a very interesting slide. So, see, we gave GnRH, we give for ARTs, we give GnRH, right? So, in the increase vaginal pH significantly above 4.5. So, when we give GnRH, estrogen and progesterone suppression occurs initially. So what will happen is decrease of the hormone and the pH increases. In endometriosis, significant shift towards immediate vaginal microflora, decrease in normal microflora. In control group, similar but non-significant shifts, significantly higher colonization of Gardnerella, E. coli in both the groups. Application antibiotic is levofloxacin, reduced growth of E. coli is seen. So coming to the treatment of ART and its effect on VMBs. So when we give GnRH, agonist treatment. So it affects the vaginal pH. Next slide, please. Effects of ART on vaginal microbiome. VMB. Hormonal influences during IVF. Significant higher CPR in the group with no microbial growth compared with these group cultures were positive for microbial colonization. Next slide, please. Effects of ART on VMB. Significantly, this is the group 2 where the CPR is lower. Ongoing pregnancy rates and implantation rates in group with positive cervical culture. Next slide, please. Effects of ART on VMB. Circulating hormones impact greatly upon the vaginal microbiome, susceptibility of the woman to the infection. Next slide. Ovarian stimulation. Using 16 srDNA deep sequencing and IVF ET. In an IVF ET, that is the highly purified FSH. Shift in the VMB in some but not all of the women. Novel bacterial strains have been found in 33% of the women screening during the treatment cycle. So ART, we have to watch for the vaginal microbiome, and if we have to screen the patients for VMB, and if any low VMB or low lactobacillus is seen in the slides, supplement them. Next slide, please. Next slide, please. Hello. Yeah. So VMB on the day of ET. This is an interesting thing. So ET is transplantation, right? ET is embryo transfer. So VMB levels and the outcome of pregnancy with ET. So effects pregnancy outcome. Women with ongoing pregnancy was characterized by top genera by some fraction. Lactobacillus species were the top species. Great care to reduce the risk of microbial transfer into the cervix during the ET by cleaning the we take these precautions, right? We clean the external os or the cervix with culture media, avoid touching the vaginal walls and external cervix with a catheter tube, screen the patient carefully before we go for ET. Next slide, please. Next slide, please. In tubal infertility. So abnormal VMB significant associated with tubal infertility. Chlamydia T are the more prevalent in bacterial vaginosis positive women. Components of an abnormal VMB ascend through the cervix, upper genital, pelvic inflammation, reduce fertility. So abnormal VMB is associated with tubal infertility or tube blocks. And the common organism is chlamydia. Next slide, please. In patients undergoing fertility treatment, bacterial vaginosis and abnormal lower genital tract microbiota was significantly more common compared to the general population. 1 in 5 infertility patients have BV. See, the incidence has increased to 80%. 1 in 5. 1 in 3 have a disturbed vaginal microbiota. BV does not affect the conception rates. This is an important point. So the infertility patients, very important to screen the vaginal flora, to know the pH of the vagina, to know if bacterial vaginosis is there. Next slide, please. Infertile patients undergoing IVF. Implantation rate was reduced in those women who were BV compared to BV negative. Difference was not statistically significant, 27.6 and 36.3% BV negative. But still that percent is quite important in IVF patients. Next slide, please. Effects of ART and VMB. Contamination is possible from vaginocervical microorganisms by needle puncture in the vagina, microorganisms that enter the endometrium from the cervix during embryo transfer could damage the developing embryo and prevent pregnancy. Next slide, please. Pathophysiology of vaginal imbalance in female infertility. Next slide. Increases concentration of the leukin factors, that is IL-1B in the vaginal environment, which triggers off the killer factors. And automatically the sperms are killed along with the vaginal flora. Trigger the generation of the reactive oxygen species, affects the membrane and integrity and flexibility, impairs the tail motion and sperm oocyte fusion. Very important. Next slide, please. That is, indirectly, it affects the implantation, fertilization, movement of the sperms, acrosomial reaction, everything is affected. Next slide, please. So now, what to do with this? If, okay, we have investigated the infertile patient, the VMB is less, right? So the pH is high. We treat the patient with the antibiotics. Now, come to the newer factor of probiotics. The hallmark of a healthy VMB is, in most women, relative abundance of Lactobacillus. Next slide. So we are coming into a pictorial form of what we read till now. That is the antral follicles, dominant follicle. We are getting an estrogen. And when this doesn't occur, ovulation doesn't occur, the lack of estrogens. And when the pH is lactic acid, progesterone is not there, estrogens are not there, glycogen is not there in the vaginal epithelia. And the lactobacillus cannot produce the lactic acid, reduce lactobacillus, and it increases the pH, which is susceptible to the infections, increased pelvic inflammatory disease, sexually transmitted diseases, PID, sperm motility, viability, everything is affected. Conception is affected. And here we have good ovulation, good progesterone levels, where the pathogens, see, addition to the pathogens, increase in the lactic acid levels, increase in the fertilization and implantation rates, high pregnancy rate, continuation of the pregnancy, and baby in the hand. Next slide, please. So we know about the antibiotics we give to the patients. So now looking to the future, a healthy vaginal microbiota can help to prevent the urogenital conditions such as UTIs, bacterial vaginosis. Therefore, studies leading to a better understanding of the VMB will facilitate the discovery of improved treatments and diagnosis for such conditions. Definitely, the recurrences of BV decreases by 80% when we use the probiotics. Next slide, please. RBM online, what fertility specialists should know about the vaginal microbiome. Key message from him. He's the director of the IV1 Madrid. He's a professor of obstetrics and gynecology at the Rey Juan Carlos University, Spain. His main research is IVF and endometriosis. He has published about 150 articles and 22 book chapters on human reproduction, endometriosis, and other topics. So the key message, Vaginal microbiome or VMB comprises composition varies between women and has the capacity to fluctuate throughout the woman's life. The abundance of various bacterial species in the vagina greatly impacts a woman's reproductive health and pregnancy outcome. The VMB may be altered by exogenous factors that may impact reproductive health outcomes. Good. Next slide, please. Looking to the future, probiotics containing lactobacillus used over a long time, an alternative to antibiotics, particularly with the persons who are risk of high infection and recurrent infections. Next slide. So according to the WHO, probiotics are defined as live microorganisms which, when administered in adequate amounts, confer a health benefit to the host. This also works for GIT infections. Characteristics of potential probiotics: acidic and bile stability, human origin, production of antibacterial, antimicrobial substances, adherence to the human intestinal cells, persistence in the human intestinal tract, clinically validated and documented health effect, antagonism against enteric pathogens, susceptible to antibiotics, safety in food and clinical use. Next slide, please. So, prebiotics. Prebiotics are compounds in food that induce the growth or activity of beneficial or good bacteria. So we have prebiotics, probiotics, both of them together called are symbiotics, right? Now prebiotics. Prebiotics stimulate the growth and reproduction of other only useful microbacteria. They maintain the pH, stimulates local immunity, remove excess mucus from the walls in the small intestine, reduces the formation of gases, suppresses the reproduction in the intestines of pathogenic bacteria. It stimulates peristalsis, improves the work of the digestive system. Next slide. So this is from the Human Reproductive Update. Same person, Koidoker et al. 2019. We therefore wanted to answer the following questions. Is the presence or absence of certain bacteria associated with failure to become pregnant after an IVF or ICSI treatment? Can the composition of the vaginal VMB be used as an independent predictor of for IVF or IVF outcome? No. Next slide, please. So what does these studies indicate? What are the answers for the questions? So our results indicate that VMB profiling using the IS-PRO technique enables stratification of the changing chance of becoming pregnant prior to the start of an IVF or IVF treatment. Knowledge of their vaginal microbiota may enable couples to make or balance decision regarding timing and continuation of the IVF and ICSI cycle. See, they cannot tell us that this couple is going to be pregnant or not, but definitely it can give us and it can tell us, should we continue the IVF or we need to treat the BV, we need to get the VMB into track and then go for IVF. Next slide, please. This is a study again, differential composition of VMB, but not of seminal microbiome in association with successful IUI in couples with idiopathic infertility. So what they have done is in the study, they have done IUI in 23 couples and vaginal swab also was taken to see how the lactobacillus is there in these couples and what are the results they have seen. Our results strengthen the potential of lactobacillus in promoting a favorable environment for pregnancy and suggest that microbiome characterization could be useful together with standard clinical and laboratory assessments in the pre-IUI evaluation of an infertility couple. As I said, see, it cannot tell us whether the couple is going to be pregnant or not, but definitely it is going to tell us, should we go ahead with IUI and the failure rates will be more when the VMB is less and the colonization is seen. Right? Next slide, please. And it is proven that the fertility rates decrease when the VMB is low. Next slide, please. Abnormal VMB babies with poor reproductive outcomes, a prospective study in IVF patients. And even the pregnancy rate, continuation of the pregnancy, abortions increase with increased colonization or decreased VMB. Next slide, please. Hello, next slide, please. Relevance of assessing the uterine microbiota in infertility. See, we were assessing the cervical or maybe basically the vaginal and sometimes the cervical VMB. A study showing that about the endometrial VMB. And it is in correlation with the VMB of the vagina. However, the study of the endometrial microbiota, as with the other low biomass microbiota, presents important hurdles because due to the small amount of the starting material, they are easily contaminated by exogenous bacterial DNA. For this reason, there is no conclusion, or you cannot say that endometrial VMB and effect on pregnancy or chronic endometritis, but definitely there is effect on the fertility issues. Next slide, please. Potential influence of VMB on infertility and ART. So have been proved by meta-analysis demonstrated by odds ratio 33.3, 95% confidence interval showing that VV affects ART results. Women with tubal factor have significantly increased BV. 40% of the patients undergoing ART have BV. Next slide. Coming to the next part of IVF, that is, we have seen the endometrium, cervix, vagina, tubes. Now coming to the implantation. ET is done. We have discussed about endometrial transfer. Now coming to the implantation. Does lactobacillus have effect on implantation? Indirectly, yes. The normal flora of the RT includes a variety of lactobacillus species, healthy supportive environment for the embryo for implantation, provides safe environment for implantation. Lactobacillus comprise 95% of the total bacteria count in the reproductive system. 4 species are seen: Crispus, Caenorus, Genserioli, and Lactobacillus gasseri. 2 main attributes of lactobacilli have been shown to play a pivotal role in shifting the balance of the reproductive tract environment for safe implantation and continuation of the pregnancy. Lactobacilli provide lactic acid, which lowers the pH and is uncomfortable for the habitat of many pathogens, including BV. Live birth rate have directly been correlated with the recovery of oxygen radicals produced by the lactobacilli from embryo, transfer, catheter trip, and inversely correlated with bacterial vaginosis. Next slide, please. Coming to the cellular physiology from a journal of cellular physiology, subclinical alteration of the cervical vaginal microbiome in women with idiopathic infertility have been noticed. Quantitative assessment and identification of specific microorganisms of the cervical vaginal microflora could increase the accuracy of available tools and the diagnosis of infertility, and also decrease abortion rates and preterm labors. Next slide, please. So this is again a study from reproductive health. Recept IVF cohort study protocol, the urogenital VMB and predictor for IVF. 300 women on the reproductive stage study were studied with IVF, with or without ICSI. The endpoint, and everyone a vaginal swab. These women and midstream urine sample and a vaginal swab were done. So the endpoints of the study are pregnancy achieved after fresh embryo transfer and within the subsequent year after inclusion, and validation of the findings will take place in addition of the cohort of 50 women of RV education. Discussion. Predictive knowledge of the microbiome profile may enable couples to make a more sustained decision whether not to continue the treatment. Unnecessary physical, emotional burden or failed IVF can be avoided. Next slide, please. Vaginal microbiome as a tool to predict IVF studies, IVF success. As I told you, we cannot confirm through the VMB levels that the lady is going to be pregnant, but it's a definitive marker to tell us when to stop and start back again. Next slide, please. To investigate the VMB influences the IVF outcome. Methods, here the 31 patients who are undergoing ART with blastocyst transfer were studied. So the vaginal VMB can influence the results of ART. The profiles dominated by Lactobacilli versus with higher pregnancy rate and achievement of pregnancy. This is a study done by BMC, published on 24th August 2019, and showed that ART is influenced by vaginal microbiome. Next slide, please. So as we have seen a study about 5 studies ago, effect of endometrial MBB and implantation success or failure. So reproductive endometrium, they have studied on all the patients whom they are doing ET, and they have taken a receptive endometrium decreases, and when the presence of non-lactobacillus dominant microbiota, means other infections are there in the endometrium, implantation rate goes down drastically. So we need good endometrial bacteria, microflora. Next slide, please. For the implantation to happen. So treatment of the bacterial vaginosis improves ART. Next slide. Fine. Thank you. Any other questions?
Thank you very much, madam, for this nice and informative presentation. Just give me 1 minute time, ma'am, to check the questions again.
Sure.
Madam, the first question is, does this probiotics can be given in all the patients prior to move for this ART?
See, ART already we are burdening them with many medications, injections and medications. Any slight sign of decrease or increase in the pH, any sign of infections, any sign of cervicitis, any sign of endometritis, or any sign of infection in the tubes, tubal blocks, or categorize the patients. Then we can definitely give probiotics as long as we are doing the ARTs. But in other factors, like, even in endometriosis, we can give probiotics, but I would prefer prebiotics. But in cases of, ovarian factors, and failure, ovarian failures, or these cases, I won't advise probiotics. Just categorize the patients.
Yes, thank you. Thank you very much, madam.
Mm.
And madam, another thing is which bacterial strain is the most important in case of this probiotic use in ART, which is recommended mostly?
All bacterial strains do cause damage to our treatment. So I need not categorize that I'm going to give probiotics for this bacterial strain. E. coli, the probiotics work wonders, basically. Chlamydia, they need definitely antibiotics coverage. And you need not categorize this bacteria definitely give probiotics. But I would recommend E. coli when you find E. coli infection, probiotics and prebiotics, definitely, prebiotics, definitely. You have to go for symbiotics.
Yes. Okay. Thank you. Thank you very much, madam, for the clarification. And I could not find any other question, ma'am. So if you allow me, then we can conclude this session, ma'am.
Yes, please.
So thank you very much, madam, once again for your valuable time and for discussing such a nice topic and very important topic, which is very pertinent to the ART also. So once again, thank you very much. And in future also, ma'am, we will look forward for some valuable session with you in our Shield Connect platform. So thank you very much, madam. Have a good evening. Thank you.
Thank you so much. Yeah.
Thank you, ma'am. Thank you very much, ma'am.
Yeah. Thank you, Yakaya.
Thank you, ma'am.
Did they grow through ante this topic kada? Did they follow? I mean, who are listening? Hello?
Yeah, one second, sir.
Madam, already participations are there in our delegate link and YouTube. So after completion of this program, they will share the data with you.
Yeah, because the topic is little difficult topic. So I'm not sure if they could follow. Is it clear for them or they have any doubts or everything is okay?
No, I guess it's okay, ma'am.
Yeah. Thank you.
Thank you.
Okay,
Very
bye.
much.
Yeah, thank you, ma'am. Thank you so